Hi, good evening. So, am I audible and visible? Audible and visible, right? Hi, good evening. Okay. Right. So, today's session is a continuation of yesterday's, right? So, what did we see in the yesterday's sessions? Molecular biology. We've been saying about replication, right? Nucleotide metabolism, furin metabolism, pyrimidine metabolism, and then about replication. So in prokaryotes and eukaryotes and the inhibitors of replication. So this is what we had covered yesterday. So in today's session, we'll continue with DNA repair mechanisms and followed by transcription in eukaryotes and prokaryotes. Right. Okay then. So this session is for MBBS Prof 1 Biochemistry. Okay, so we will be starting off with today's session. So here are two case-based questions that have been asked. One is the proband was a 40-year-old male was diagnosed with poorly differentiated adenocarcinoma. Now, a family counseling revealed that the proband had 5 family members with colorectal cancer. So here is the point that you need to concentrate, which is a colorectal cancer that was diagnosed before 45 years of age, right? That was diagnosed before 45 years of age. So just give me 1 second, please. Yeah, I'm sorry, guys. Right. So we will start now, right? So this is a case of colorectal cancer. So all the family members were diagnosed with colorectal cancer before 45 years of age, right? So as hereditary non-polyposis colon cancer is suspected, all family members were counseled that this is caused by a defect of DNA repair. So everyone older than 25 years has to undergo regular colonoscopic examination. So they have given us the diagnosis itself. Yes, so HNPCC. What are the errors that can happen in a DNA? Yes, tell me what are the errors that can happen in a DNA and what is the most common error in a DNA? Which repair defect causes HNPCC and describe in detail about the repair mechanism. So we'll be discussing about all the DNA repair mechanisms. Okay. Right. And similar question in which this is a question in which they had again asked a 42-year-old male was diagnosed with poorly differentiated adenocarcinoma. And family members, again, five family members with colorectal cancer were diagnosed before 45 years of age. So a similar case, right? So again, they have asked you what is the repair defect here? Yes, very good. So what is the repair defect here? And we must describe in detail. So these three tables are very important tables that we need to memorize. Okay. Right. So what are the DNA damaging agents, first of all? So what are the DNA damaging agents here? So we have ionizing radiations like X-rays and anti-tumor drugs. Yes, that can cause DNA lesions, especially your double-stranded breaks. Okay. So it causes break in both the strands of your DNA. And DNA repair pathways. What is a DNA repair pathway when you have a double-stranded break repair? It is non-homologous end joining and homologous recombination. Okay. Similarly, you have UV light or chemicals. UV light or chemicals, what kind of damage does it cause on a DNA? It causes pyrimidine dimers, right? Instead of the two strands held together by hydrogen bonds, the adjacent nucleotides will form bonds here. That's what is a dimer, right? And it is a bulky index, so pyrimidine dimers will be formed. And how do you repair this? It is by nucleotide excision repair. Then oxygen radicals, free radicals, hydrolysis, alkylating agents. How do they cause damage to a DNA? They cause damage by adding a base, by adding some other base other than the normal base. So abnormal bases are getting added here, like 8-oxoguanine base is added, or A basic sites, you don't have a base there, or single stranded breaks. So this can be repaired by base excision repair. Then replication error. So replication, as and when replication is happening, the replication error is common. So it can be corrected by mismatch repair. Okay. So this is how it is. So during, bases will mismatch. Yes. So bases will mismatch. Like A has to pair with T. In that case, A can pair with C or something else. So it is mismatch repair. The Watson and Crick base pairing rule is not getting followed here in case of replication error. Right. So these are the different DNA damaging agents, and these are the different DNA lesions that can happen because of these damaging agents. And these are the DNA repair pathways. So what are the pathways here? Mismatch repair, base excision repair, nucleotide excision repair. Then you have non-homologous end joining or homologous recombination. Right? So the same thing is given here. These are types of damage that can happen in a DNA. So single base alteration or two base alteration is in case of UV light exposure, and the formation of pyrimidine dimers. Then alkylating agents can cause cross-links, and chain breaks, single-stranded breaks or double-stranded breaks, and also cross-linkages can happen. So these are the types of damages that can happen in a DNA. So as we had seen. Now, these are what are the diseases that can happen because of this DNA damage repair defect. If the repair defect, if the repair mechanism is not happening, what disease can you get? If there is a defective DNA mismatch repair, That is what is HNPCC. Yes, so as in this case, it is the HNPCC, right? So this is what we are dealing about with this case. So HNPCC is a defect in the mismatch repair. Now, what is a defect in base excision repair? It is MUTYH associated polyposis. Then nucleotide excision repair defect, xeroderma pigmentosum. Again, you may get case-based question based on the xeroderma pigmentosum. So you think about nucleotide excision repair. Then cocaine syndrome, also trichothiodystrophy. Then defective homologous repair, AT-like disorder, and you have a Nijmegen breakage syndrome, Bloom syndrome, Werner syndrome. Yes, you can just memorize these. Breast cancer susceptibility 1 and 2. And then Rothman-Thompson syndromes, of which you will know Bloom syndrome. It's commonly you encounter. Then breast cancer, BRCA1, BRCA2 mutation and all. Then defective non-homologous end joining repair defect. SCID. SCID is the main here, and radiation sensitive SCID. So severe combined immunodeficiency disease. All right. So these are the diseases that can happen whenever there is a defect in your DNA repair mechanisms. Right. Next, we'll move on to what are the different. So, what is the repair here? What is the repair here? As we have seen, it is the mismatch repair defect. Right? It's the mismatch repair defect. Now, what are the different errors that can happen in this in a DNA that we have seen? What is the most common error? Most common error is what is the replication error, because replication is happening on and on, right? It's happening frequently. So, replication error is what is the most common error. Now, let us discuss about the various types of DNA repair mechanisms. So, first one is the mismatch repair. Yes. So, first one is the mismatch repair. So, mismatch repair, how do you identify? First, here, what is happening here is mismatch repair. So, what happens here? We have a daughter strand. Yes, you have a daughter strand here and it's a parent strand here. So a parent strand, how do you identify a parent strand? A parent strand is identified by the presence of methylated adenine nucleotides. So these adenine nucleotides are methylated. So that's how you identify a parent strand. Now parent strand, if you identify, you should know that the parent strand is the correct strand and whatever the daughter strand has to be corrected. Okay. So as you can see, here is the T that is paired with G, right? So G and T are not hydrogen bonded. Is it not? G and T are not hydrogen bonded. So this is a mismatch. So here the base pairing is not proper here. So now what I should do? Which strand do I take it as a correct strand and repair the other? Yes. So I will take this parent strand in which there is methylated adenine nucleotides as the correct strand. Yes, very good. As the correct strand, and I will correct the opposite strand, right? So here I have T in the parent strand. So I will remove this G, yes, and I will add the A. Yes, I will add the A. That's how I should correct. So who will help in this? MUT proteins are going to help us. So this MUT proteins are going to recognize this mismatch, and also it will identify the parent strand that is methylated, right? And then what it will do is it will cleave. It will, it will remove that segment which is wrongly attached. So this G is what is wrongly attached. So that I will remove. After removing, who will come? Polymerase. Polymerase enzyme will come and I will add the correct nucleotide there. Which is the correct nucleotide? A is the correct nucleotide. So the polymerase will add the adenine nucleotide on opposite of this T. Yes. After which the ligase is there, which will seal the nick by creating phosphodiester linkages. So that's how this is happening. So this is about the mismatch repair. So I will identify. So how you identify the mismatch, and MUT proteins are what are important here in identifying the mismatched base, and then it will remove it. Then polymerase comes and adds the correct nucleotide. Then your ligase is what it will seal the nick, right? So this is what is about your mismatch repair. Now look at this. Look at this. What has happened here? Here is the pyrimidine dimers has happened, right? So what is this? Pyrimidine dimers are being formed here. Yes, so this is what we had seen here, right? So pyrimidine dimer. So how was it repaired? It's by nucleotide excision repair. Yes, so this is nucleotide excision repair. Yes. So in nucleotide excision repair, so what do you find here? You have this pyrimidine dimers that have been formed. So instead of two strands getting, the nucleotides on opposite strand has to be hydrogen bonded. Instead, the adjacent nucleotides are getting getting bonded. That is why you form this dimer. So usually pyrimidines are what are forming dimers, thymine, thymine, dimer. So that is why you call a pyrimidine dimers are more common. So first, what I will do, I will use a UV specific endonuclease or otherwise UVR ABC exonuclease. Okay, it's not exonuclease, it's exonuclease. So this exonuclease will now remove this dimer. Yes, remove this dimer along with few nucleotides on either side of the dimer, right? So it will, or the first step, what it will do is it will first create a nick, few nucleotides away from this dimer. Yes, so it will create a nick, few nucleotides on either side away from the dimer. After which this is being removed. So this oligonucleotide as such will get removed. Then you have this DNA polymerase that will start adding the nucleotides, correct nucleotides, and then DNA ligase that will seal the nick by the phosphodiester bond. So now this DNA is corrected. Okay. So this is how happens in nucleotide excision repair. So first a pyrimidine dimer is formed. I will use a UV specific endonuclease or an exonuclease to form a nick on either side of the dimer, few nucleotides away from it, and then the oligonucleotide which is damaged is being removed. Then DNA polymerase will fill the gap. DNA ligase will seal the nick. So this is what happens in nucleotide excision repair. So this is very easy. Next is what is the base excision repair. Next is what is the base excision repair. So where this base excision repair mechanism comes into play, wherever there is addition of wrong bases have been wrong bases have happened, right? So look at this first, the DNA, this is the DNA there, two strands of DNA. Now spontaneous deamination of the nucleotide, this C nucleotide has happened. So whenever cytidine gets spontaneously deaminated or deaminated cytidine is what is called as the uracil, right? Now what happens? Uracil is having, it's in the DNA. So in DNA, do you find uracil? No, right? In DNA, there should not be any uracil because when you have this uracil is found only in RNA. So when you have this uracil, then the bond will be very weak and it will, the DNA will not have its stable structure. So uracil becomes, uracil is there in this site. Now what should I do? I will use a uracil N glycosylase enzyme to remove this base. Yes, so I've just removed the uracil here. Now as the uracil is removed, now this sugar phosphate backbone is there. Only the base I have removed. Okay, so that is this kind of a space is called as the A basic site, or here this is the pyrimidine. So I will call it as a pyrimidinic site because the base alone is removed. Here the sugar phosphate backbone is retained. Next, I will use a pyrimidinic endonuclease. Okay, pyrimidinic endonuclease that will create a nick at that area. Then deoxyribose phosphate lyase that is going to remove the deoxyribose and the phosphate. Then I will bring this CTP because C is what I have to add in this area. So that will be added by DNA polymerase and DNA ligase will seal the nick. So now the correct cytidine has been added on to the DNA. So this is how it is. The base excision repair is happening. Yes. Is it fine now? Base excision repair, what has happened? When your cytidine gets spontaneously deaminated, you find uracil. So this is just one example. So as in the previous slide, we had seen many examples, right? So this is like, We have seen these kind of an example of 8-oxoguanine is getting added. So these things also you will just remove the base with that specific enzyme glycosylase, and then you will uh, bring this apyrimidinic endonuclease. It is going to form the NIC. Then deoxyribose phosphate lyase that is going to remove the sugar and phosphate. Then the correct nucleotide is brought by this DNA polymerase and is getting added. Then ligase will seal the NIC. So this is what is going to happen if a wrong base is added into the DNA. Yes, so this is what is base excision repair, right? So in all these three mechanisms, what we have seen is we have this parent strand intact. One strand is intact. So this parent strand, we were assuming that this parent strand is correct, and we are correcting the other strand, right? So in this cases, one strand is good, the other strand only there is that wrong base which is there, or nucleotide or break or whichever has happened is only in the daughter strand. In the parent strand, we have it to use it as a template. But what if both the strands of the DNA are getting broken down? That's what is called as the double-stranded break. Right? So double-stranded break. Now, if you have encounter a double-stranded break, both the strands are getting broken. So which one will you use as a template? That is very difficult to understand, right? So here what will happen? We have two mechanisms. One is non-homologous end joining and homologous end recombination, right? So non-homologous end joining means it just as the this is the DNA and it is being broken. I will just attach these two together. Okay, so I'm not going to correct it. I'm just going to attach it instead of leaving it broken like this. I'm just going to attach it. That's what is non-homologous end joining. Homologous end repair means what? Homologous end recombination means what? There are sister chromatids, right? So the homologous pair of chromosomes. So the homologous pair of chromosomes will have similar arrangement of nucleotides. There can be exceptions, but still similar arrangement of nucleotides is there. So suppose this is one DNA which is being broken. I have its homologous pair inside the cell, so that will come closer, and this homologous pair will now act as a template and correct the broken strand, right? So that is what is homologous end recombination. So let's see these two mechanisms now. So first, non-homologous end joining. This is by Q helicases. Yes, Q helicases and protein kinases. Q helicases and DNA protein kinases. So look at this. This strand is now broken. So both the strands have been broken here. Now these Q helicases and protein kinases will go to the ends of the broken ends. Okay, it is going to the ends of broken ends of the DNA. So it is going and sitting in the broken ends of the DNA. Now it will trim. Okay, some sort of trimming is going to happen. Yes, so these two are going to trim these broken ends. After which what happens is, so once I have trimmed some of the bases, right, what it will do is both the strands are going to search for its complementary nucleotide, the other strand, right? So suppose these are the two broken strands. Yes, so both the strands are going to find its way, and it is going to approximate each other, and it will in search of complementary nucleotides between these two strands itself. Once it finds a complementary nucleotide, it will just get attached like this. And the remaining portions, you have these remaining portions, right? These remaining portions will now be trimmed and removed. So you will have, I will just, you've just approximated. Whenever you find a complementary base, it will stop and the remaining ends are getting cleaved. So this is how I'm just attaching and joining these two broken strands, right? So look at this picture here. So once the trimming has happened, what is happening is it is just getting approximated. Okay, so both these double strands are getting approximated. Now it will find its base pair. Okay, find its complementary pair here. After it finds its complementary pair, after it finds its complementary pair, now the ends, these remaining ends are going to be trimmed off and the strands are getting corrected. So this is how I am correcting it. But what's the difficulty here? Yes, there is loss of nucleotides. Is it not? There is loss of nucleotides here. So there is loss of portion of DNA here, and hence loss of the genetic material, which is of course dangerous, right? So that is why this is highly error prone. But instead of leaving the DNA broken, I'm just I'm just attaching and sealing it. So that is the one good thing that I'm doing in non-homologous end joining. And this will happen in G0 or G1 phase of cell cycle. Understood? Next is what is the homologous end recombination. So are you understanding this? Yes? If yes, just leave a message. Next one, we'll move on to the homologous end recombination. Okay, so here what happens again? This is not tough. Okay, just try to understand. It is easy. So this homologous end recombination, it is especially for a double-stranded break repair mechanism. Here, my homologous pair is going to come closer. Okay, so this is a so the red one is what is the broken DNA strand, and blue one is what is this homologous pair. So now after it is broken, after the strand is broken, the homologous pair is coming closer, and then you have something called as the mRNA complex, which is a complex of proteins that is going to get itself attached to the broken ends of the DNA. Okay, broken ends of the DNA, and it will cause some trimming. That is what is called as the end resection. So end resection of this broken ends are going to happen. How does it resect or how does it trim? It trims in such a way that it leaves off 3' overhanging ends. Okay, it leaves off 3' overhanging ends. So after which what happens? So here is the blue color one is the homologous template. Yes, that I am using it as a template now. Complementary to that is what I will correct the broken strand. Okay, let's see how this is happening. So I have this end, overhanging end here, right? So this overhanging end is going to invade this homologous strand. So before that, what will happen? This homologous pair will unwind itself, okay, and opens up. The homologous pair is going to unwind and open up so that the broken strand get invade between the two, and it can use one strand as its complementary strand. It can synthesize nucleotides complementary to it. Is it not? So this is how this overhanging end is going to invade the strand, this strand invasion and branch migration. So this I have it as a template. So complementary to this homologous template, I will synthesize or add nucleotides here. So thereby this red strand, which is the broken strand, is getting corrected, right? So similarly, the other red strand also is getting corrected by using this blue strand as its complementary strand. The blue is what is the homologous pair. So as its template, okay, blue as its template, it is going to correct this red one. So this is how both the strands of this double-stranded DNA are going to get repaired, right? So now, as this is growing inwards and correcting itself, So finally, what do you find? It's invading and correcting itself. Understood? So now what happens? You have these cross junctions that have been formed here. These cross junctions are what are called as the holiday junctions. Okay, holiday junctions. So this invading, this opening up of this homologous template, this looks like a D, is it not? That's what is called as a D loop that is formed after the homologous strand is getting unwound. So now let's see the step one by one. So this is how it is. First is homologous end recombination is a double-stranded break repair mechanism. Homologous pair will come closer. End resection is happening with the help of this mRNA complex. Then 3' overhanging ends will be formed. Then what is happening? Strand invasion, branch migration is happening. Here, since this homologous template is unwound at that area, D loop is getting formed. Then as the strand invades and it adds the nucleotides complementary to that of the homologous strand, and in that process it forms these cross junctions which are called as the holiday junctions, after which I need to cut off this holiday junction. So I will remove the holiday junction so that you will or release the holiday junctions or the cross links, and thereby you get the you this is released. Okay. So homologous pair also, it's not like the complete glue, right? So there can be shifting of these nucleotides or these gene segments or these DNA segments here, right? So this is again happening, but both of the strands are corrected and filled up. Yes. So DNA polymerase is what is adding the nucleotides. DNA ligase is what is sealing the nick as usual. So this is what is about the homologous and recombination. Okay. So is it clear? Is it clear? Okay. So next we'll move on to the next question. So with this, we are completing this DNA repair mechanism. So I've explained you all 4 types of DNA repair mechanism. Yes, so this is how you would learn, and with the help of picture, just start, write it like a flow chart. Okay, so that's how you will present this question. Right. So next we'll move on to the next. These two, we have seen in the early session, but since they have asked this as a separate question, I've just concentrated. So mechanism of action of methotrexate and actinomycin D as an anti-cancer drug. Here, methotrexate is an anti-metabolite. You can also call it as an anti-folate. Okay. So look at this. Look at this picture. This reaction is very important. That's why I've just included. So DUMP, yes, DUMP becomes DTMP, uracil to thymidine, with the help of thymidylate synthase enzyme. For this, you will need N5N10 methylene tetrahydrofolate. So when this gets used up in the reaction, it becomes dihydrofolate. Now you need to regenerate the N5N10 methylene tetrahydrofolate, for which this will now become tetrahydrofolate. Yes, and this is the enzyme dihydrofolate reductase enzyme. Then this becomes N5N10 tetrahydrofolate. This is a pyridoxal phosphate dependent reaction. In this reaction, serine becomes glycine. Fine. So we are now concentrating on DHFR. So DHFR, if it gets inhibited, what is happening? This reaction will not happen. So DTMP will not be formed here. Yes. So that is a problem it's not a problem because we are using this as a drug, anti-cancer drug. So since DTMP synthesis is getting affected here, the cell will not have the adequate basis for the nucleic acid formation. So that's how it is reducing the DNA RNA synthesis as well as the protein synthesis. So cell cycle also gets arrested here. Understood? So this is what happens. Next is actinomycin D or dactinomycin. Now this intercalates between guanine cytosine base pairs in DNA. So this is how it intercalates. So this is a normal DNA. So here in this, what is happening? An intercalating agent is going and sitting between these bases, right? So as it sits in between the bases, the structure itself is collapsed here. So that will prevent the mRNA synthesis. It blocks the RNA polymerase, inhibits transcription. So in larger quantities, it will also inhibit replication. Understood? So this is the mechanism of action of methotrexate. So one more thing I just forgot to tell here. So non-homologous end joining, we saw it is more error prone compared to homologous end recombination. That's the difference you must know here. So this is less error prone. Okay. So in which phase, other phases of cell cycle, this homologous end recombination will happen. So this is one more point that you can know. Right. Next, we will move on to transcription. Right? So write in detail about the initiation, elongation, and termination of transcription and give an account of post-transcriptional processing. So this is everything they have just asked. So while learning itself, we will learn the difference between prokaryotes and eukaryotes. Okay. So prokaryotes, let us see for in this first slide, it is there. I have just mentioned about the requirements of your transcription. So what all do you need for a transcription? First one, you will need a template strand. Right? First one, you will need a template strand. So in both prokaryotes and eukaryotes, you will need a template strand. Now, what is this template strand? So among the two strands of DNA, one strand is called as the template strand and the other strand is called as the coding strand, right? So mRNA is synthesized complementary to this template strand. Okay, mRNA is synthesized complementary to this template strand. And this coding strand mRNA will be looking similar to this coding strand with the exception of U in place of T. Yes, is it not? That's very obvious. So template strand, complementary to the template strand is what is mRNA will be synthesized. Now, what is coding strand will look similar to this mRNA instead of, meaning, yes, so in uracil alone, you will find. Okay, so that's the difference here. Instead of thymine, you find uracil. So that's what you must understand. So coding strand will look similar to mRNA except for the uracil. So this is about the template strand. Next is DNA RNA polymerase is what is the enzyme that is necessary for transcription. So what is this RNA polymerase? Yes, what is this RNA polymerase? In case of eukaryotes, I'm telling you, this is in case of, in case of prokaryotes, in case of prokaryotes, the RNA polymerase looks like this. So it will have two alpha subunits, then your beta and beta prime subunit, then you will have omega subunit and a sigma subunit. Okay, so this is a core enzyme. Along with the sigma subunit, you will call it as a holoenzyme. So what are the different functions of these subunits? Alpha subunit is necessary for enzyme assembly. And sigma subunit is the most important one because that is going to identify and bind to the promoter site. Okay, that is going to identify and bind to the promoter site. Beta subunit is what can bind magnesium ions catalytic subunit. And it is the catalytic subunit. It has the 5' 3' polymerase activity. Beta prime subunit is what is for template binding and omega subunit is for again enzyme assembly. So alpha and omega is for enzyme assembly. Sigma is the important subunit. It's going to identify and bind to the promoter site. After that only transcription can happen if the promoter site is getting identified, right? So beta is catalytic activity. It has a polymerase activity and beta prime is again template binding. So this is what is the RNA polymerase in case of prokaryotes. Okay. Next is RNA polymerase in case of eukaryotes. What do you have? 1, 2, and 3. You have 3 types of RNA polymerases. 3 starts with T. So it is for tRNA synthesis. 2 is mRNA. That's what we are going to learn now. mRNA synthesis. And 1 is looking like R. No, it's rRNA synthesis. So above all, what is transcription? Transcription is a process by which you are converting the DNA to an mRNA. Yes. So DNA to an mRNA. So not just mRNA, any RNA. That's what you have to understand here. So first of all, DNA will give you a primary transcript. Okay, it will give you a primary transcript. So just the template strand, complementary strand is getting formed. That is what is the primary transcript. This primary transcript will undergo post-transcriptional modification and then it will form this mature mRNA or mature RNA. Yes, so this is the process that you have to know. Next, so we need what are the requirements? Template strand, RNA polymerase. These both are over. Then you need the promoter site. Yes, you need to understand about the promoter. So what is this promoter here? What is a promoter? Here in promoter site, as you can see in the coding strand, that is you have the TATAAT. Is it not? That is what is called as the data box. So in case of prokaryotes, this data box is called as Ribnow box. Okay, data box is called as Ribnow box. That is present minus 10 base pairs ahead of this transcription start site. So the transcription start site is denoted as +1. So ahead of this transcription start site, you have this, you have the data box or the Ribnow box, that is 10 base pairs ahead of this or upstream of this transcription start site. Next is upstream of this transcription start site, 35 base pair sequences. You have another consensus sequence, which is this. Okay, you have this consensus sequence. This is again called as the -35 base pair. So what is this consensus sequence? Consensus sequence means it's the sequence that is present at that particular position, be it any transcription start site. So any transcription start site, 10 base pairs upstream of that is going to be data box. 35 base pairs upstream of that, there is going to be this -35 base pair sequence, that particular sequence. Okay, so that sequence is not going to be differing. That is what those kind of sequences is what you will call it as consensus sequence. Okay, so this is what is the Ribnow box and then -35 base pair sequence in case of prokaryotes. So this is what is the promoters in case of prokaryotes. So how is this mRNA getting synthesized? From this transcription start site, this mRNA is produced. In what direction it is getting produced? It is produced in the 5', 3' direction. The mRNA is synthesized. All right. In eukaryotes, what is happening? In eukaryotes, also you have promoters, but not just promoters, you also have regulatory elements. That's the difference between prokaryotes and eukaryotes. So not just promoters, you also have regulatory elements. Okay. So here you can see that this is the, this is the +1, which is the transcription start site. So you have this data box here, which is called as the Hogness box in case of eukaryotes. Hogness box. Yes, what is this data box in case of eukaryotes? Just keep typing in the chat box whatever question I'm asking. Okay. So Hogness box. And then you have promoter proximal elements. These are the this cat box and GC rich region. Okay, cat box and GC rich region. These are the promoter proximal elements because these proximal regulatory elements are situated just nearer to the data box or nearer to the promoter, right? And then you have something called as this INR and DPE. Okay, INR and DPE. This is the initiator sequence and downstream promoter element. So these instead, if at all this data box is not present in any site, these INR and DPE can function as the promoter. So that is what is the function of INR or the initiator sequence and the downstream promoter element. Okay. So if data box is lacking, then it may be considered as the promoter site. Now, this is the direction of the formation of this mRNA. Yes, in the 5' to 3' direction, starting from the +1 or the transcription start site, right? So what is this proximal promoter elements, which are situated proximally to the promoter, which is the GC rich region and the cat box. And you have distal regulatory elements. Distal regulatory elements are the enhancer and repressor elements. They are not situated just nearer to the promoter. It can be situated anywhere in the DNA segments, right? It can be situated anywhere. But as a 3D conformation, the length of it can come closer to the promoter site. Yes, or the transcription start site. Thereby, it is enhancing or repressing the transcription. So that is what you must know. That's why they're called as distal regulatory elements. So what are the different? So you will have promoter as well as regulatory elements. So this regulatory elements we have seen proximal and then distal, right? So you also have transcription factors which are necessary to increase the transcription or stimulate the transcription. So What are, one is basal components. We saw you need RNA polymerase 2 for mRNA synthesis. Then you have Tata binding protein. Then you have transcription factor 2A, B, 2B, 2D, 2E, 2F, and 2H. So you have these many transcription factors, right? These are all general transcription factors that are necessary for this, for the transcription. Then you have co-regulators. Yes, you have co-regulators. You have the Tata associated factors, and then you have mediators, chromatin modifiers, chromatin remodelers, so many such proteins you will need. Then activators, these are special transcription factors like SP1, specificity factor 1, ATF, CTF, AP1. So these are all different proteins that are activators. So basically, these are the three classes of transcription factors that are involved in the mRNA gene transcription. So these many proteins you will need for the transcription to happen in case of eukaryotes, right? Next is how does the initiation, before initiation, I want a complex to be formed that is going to initiate the transcription. That is what you called as a pre-initiation complex. Here, pre-initiation complex will be this promoters. You just need the promoters to help you in the pre-initiation complex. Your RNA polymerase, if the sigma subunit is going to identify this promoter, it is going to attach to the promoter, and from there the transcription is going to happen. So that much is in case of prokaryotes. But in case of eukaryotes, you can see that you will need this a core promoter that's present. You will need not just the RNA polymerase, you will need the transcription factors also, right? So RNA polymerase, just like that, don't identify the promoter. That's the difference. Here in prokaryotes, RNA polymerase will identify the promoter and it will go and sit. Whereas in eukaryotes, the RNA polymerase will not identify, it has no subunits to identify the promoter. That's why you have this general transcription factors, that is especially the transcription factor 2D. That's going to come to the promoter, then it will recruit your RNA polymerase, other transcription factors that will be recruited. Right? So that's how it is. So this is what is the pre-initiation complex in case of eukaryotes. Right? And similarly, how does this enhancers and repressors bind to the transcription factors? These in special transcription, these are special transcription factors that we saw, right? Transcription factor 2D, 2A, 2B, these are all general transcription factors. SPF, CTF, these are all special transcription factors. So look at that, it has a DNA binding domain, and also it has another domain which is free, to which these enhancer elements can go and bind. Where are these enhancers? On which is very away from the promoter site. Yes, it is going to be very away from the promoter site, but it can closely come and approximate to the promoter site, and that's how it enhances or represses the transcription process by binding to your transcription factors. Is it clear now? Yes. So this is what happens in case of eukaryotes. So you will need all these general transcription factors, special transcription factors, enhancers, repressors, mediators, so much of protein that you will need in addition to the RNA polymerase 2 for the formation of the pre-initiation complex, right? Next is the process of transcription, that is initiation, elongation, and termination. This is going to be the same in both the from both the eukaryotes and prokaryotes. But you see, first what happens is template binding and close complex formation. So this is the DNA here, right? So here is P is the promoter. P is a promoter site. To the promoter site, what is binding? Your RNA polymerase is binding, right? RNA polymerase as it binds, it will cause local unwinding of the DNA. Yes, so the DNA has to be locally unmarked. So that's when the template strand will be free. So mRNA can be synthesized, is it not? Unmarked. Then ATP is there, nucleotide triphosphates are getting added up. What will happen? The first nucleotide that can get added up is going to be your adenine or any purine most commonly. And that will adenine trinucleotide is what will be added. Okay, trinucleotide phosphate what will be added to the first, after which N is any other nucleotide. So here in the promoter, the RNA polymerase is sitting. This RNA polymerase has this a polymerase activity, is it not? So what it will do is that it will start adding up the nucleotides one by one, complementary to the template strand. So first nucleotide, usually it is going to be adenine or any purine, and then followed by other nucleotides. So to about 10 nucleotides sitting in the place, the RNA polymerase sitting in the place of the promoter, it is going to add about 10 nucleotides. After which what happens? This RNA polymerase will now move away from the promoter and it will go to the, and it will start from the transcription start site, right? So that's what it will happen. That's what it will. So that is what is called as the promoter clearance. Okay, that's what is called as the promoter clearance. So this, after this promoter clearance, the chain gets elongated. So here only, exactly starting from this point only, the mRNA is actually synthesized. So here it will start adding up nucleotides that are complementary to the template strand. So all of the nucleotide triphosphates are there. So we know that nucleotide triphosphates, when it gets added up, nucleotide monophosphate will be incorporated in the nucleic acids, right? Then what happens? Chain termination signal, when it has been encountered, the transcription will stop, and then the mRNA is released, RNA polymerase is released, and this DNA is released. Okay, this is what is the transcription cycle. Okay, this is what is the transcription cycle. The same thing will happen even in eukaryotes, but what's the difference here? We are just having this RNA polymerase, that itself having this promoter binding ability and also polymerase activity. But here in eukaryotes, that will be done by separate transcription factors. That's what you have to understand, right? So complementary to this, in what direction? 5' to 3' direction, this transcription process is going to happen. The mRNA is going to be synthesized. Clear? Next is termination. Termination can happen in row dependent and row independent manner. You will see it in the next slide in case of prokaryotes. In eukaryotes, it will cleave the primary transcript. Who will cleave? RNA endonucleases will cleave the primary transcript. Just 15 bases, 3' of a consensus sequence which has this A and U. Clear? So this is the termination in case of eukaryotes. All right. Now, what is this row dependent and row independent? Row is a protein that is going to cut your mRNA, or it will release the mRNA from the DNA. Okay. So this is a direction of transcription. Now, as you can see in the coding strand, yes, as you can see in the coding strand, what is there? There is one region which is rich in GC, right? And then after a few nucleotides length, there is a region again rich in, GC, right? So these two are actually looking complementary to each other, is it not? Similarly, you see the template strand. We have this CGGGCG. Here, few nucleotides away, I have again CGCCCGA, right? Followed by which I have AAA, poly A, right? So now when my the template strand, when a complementary, that is the mRNA, is synthesized, what will happen? These two areas will be formed as self-complementary sequences. Is it not? These two areas will form as self-complementary sequences, as you can see here. So this self-complementary sequences can bind together, right? But on the end, what do you have? A and U here. Because at the end, you have this AAA sequence. So because of which the UUU is what is formed in the mRNA. Okay. So now this AU bond is weak. Is it not? AU bond is weak. So because of this weak bond, what happens? It just cut. Okay. So if row independent manner, AU bond is weak, so the transcription starts at that point. Okay. So a hairpin loop, which is self-complementary to each other, is formed in the mRNA. And then at the end of this loop, you have this A and U, which has a weak binding. So that's how the transcription is terminated there. This is row independent fashion. Row dependent means you have a row termination factor that is going to interact with the paused polymerase and it will induce chain termination. So that itself will stop the transcription. So this is how termination happens in eukaryotes in row dependent and row independent manner. Right? Next. What is the difference? One unique feature of eukaryotic transcription is what? It is regulated. Yes, it is highly regulated. So you have this euchromatin structure and heterochromatin structure. Yes, so changing of this euchromatin to heterochromatin and vice versa is what is called as the chromatin remodeling. Okay, chromatin remodeling. Heterochromatin is a highly condensed form and there is transcriptionally inactive. Highly condensed means what? The histones and the DNA are highly tightly packed. So you don't have space for transcription factors to come and initiate the transcription. So that kind of a structure is what is heterochromatin and it is going to be transcriptionally inactive. So whenever you don't want transcription to happen, it will stay in the heterochromatin structure. When you want transcription to happen, what will happen? Histone acetylation can happen. Yes, histone acetylation, what does it do? So basically the histones are all actually positively charged with more of lysine residues. And DNA, you know, it's negatively charged. So that's how histone and DNA will be binding towards each other. If I acetylate histone, the positive charge on the histone is going to be reduced now. So the binding of this DNA to histone is going to be reduced. So there will be unwinding. Okay, so that's how it's a decondensed form. And this unwinding, if it happens, transcription factors can come and initiate transcription. So it is transcriptionally active. That form is called as a chromatin or a chromatin structure. All right, so this is a unique feature which is present only in the eukaryotes. Then inhibitors of prokaryotic transcription, you have rifamycin and actinomycin B. In case of eukaryotes, you have alpha amanitin that you find in the mushroom poisoning. Okay, and post-trans, so whatever DNA is there, you form an mRNA. Is it not? This mRNA is not directly formed. What did I tell you? A primary transcript is formed and this primary transcript will undergo post-transcriptional modifications. That is what is 7-methyl-guanosine capping, polyethylene splicing. These three processes are post-transcriptional modifications after which you form this mature RNA and the mRNA. But in case of prokaryotes, what is happening? Post-transcriptional modifications. No, mRNA, just the mRNA which is formed out of this, process is mature. So the transcription and translation will go hand in hand. There is no break in between, as in case of post-transcriptional modifications. The transcription and translation will go hand in hand in case of bacteria or in your prokaryotes. So in eukaryotes, we have post-transcriptional modifications. So let's see that. So first one is what? 7-methyl-guanosine capping is what is the first one? 7-methyl-guanosine capping. 7-methyl-guanosine triphosphate gap. Right? So this is what is the starting of your mRNA, which is the first base. So here there is a triphosphate linkage, which is this 5' triphosphate linkage. So as you can see, the phosphate group in the 5' area, here also it's 5' area. So 5' triphosphate linkage is what is formed, by which 7-methyl-guanosine triphosphate is getting attached to your head of the mRNA. Okay, so here what enzyme do you need? You will need guanylate transferase. Okay, you will need guanylate transferase, and the methyl donor is going to be S-adenosyl methionine. You have this enzyme guanine 7-methyl transferase enzyme. So these two enzymes you will need. Similarly, this is 7-methyl-guanosine capping. Next, you have this polyethylene. Poly A getting added up, right? So how does it get added up? It will identify a polyadenylation signal sequence, that is AAU-AAA, followed which multiple A's are getting added up. This is polyethylene. So for polyethylene, what enzyme do you need? You will need polyadenylate polymerase. Okay, you will need polyadenylate polymerase, right? So you will need these two enzymes for the 7-methylguanosine capping and poly-A tailing. What is the use of these two capping and tailing? It will prevent the mRNA from getting attacked by nucleases. Also, it will guide the mRNA towards the ribosome for protein synthesis initiation. Clear? So these are the two post transcriptional modifications. Similarly, the another process is what is splicing. What is splicing? The primary transcript will have coding regions along with non-coding regions. Coding regions will be called as exons. Non-coding regions will be called as what? Introns, right? So I want to remove these non-coding regions. I just want these coding regions alone. So attachment of exons, removal of introns is what is called as your splicing. So how does this happen? First, as you can see, this is exon 1, exon 2. Okay, look at this, follow this picture carefully. Exon 1 and exon 2, which is separated in between by an intron. In an intron, you have this A, which is adenine, which is a branch site. In this, now what happens is that you have snRNPs which are necessary for the splicing. snRNPs are the small nuclear ribonuclear proteins. You may otherwise call it as SNRPs. Okay, so these proteins you will need it for splicing process. Right now, this area is called as the 5' splice donor site of this intron. And this end of this intron, you call it as the 3' splice acceptor site. Now, with the help of ATP, it's an energy requiring process. The 2' OH of the branch site, you have this 2' which is free, right? This is an mRNA. In mRNA, in RNA as such, you have this 2' hydroxyl group which is free. In DNA, what happens? The O will be removed. That's why you call it as a deoxy. Here in RNA, you have this 2' OH which is there. So this 2' OH can attack the 5' phosphate of the splice donor site of this intron, and it will form a 2' 5' phosphodiester bond. So there is this formation, lariat structure which is formed. Now what I will do? I have this free 3' end, free 3' end of this exon that can go and attack this 5' phosphate of the splice acceptor site, and thereby it will remove this lariat structure and it will form a bond between this exon 1 and exon 2. After the lariat structure is removed, all the snRNPs are removed, and then the exons get attached together. So that's what we call it as the spliced RNA, okay, or the mature mRNA, where you have only the coding regions. Clear? This is the splicing process. So there is something called as the alternative splicing. What is this alternative splicing? Suppose there is exon skipping or retaining of introns that you will produce two different protein components, right? So how is that possible? So look at this. After formation, you have this exon, intron, right? So normally splicing, I will just add the exon 1, 2, 3, 4, and I will form a mRNA that is going to get translated and form a protein. But if a protein, if I just skip this fourth exon, only exon 1, 2, 3, I'm just adding it up, then that will produce a different protein. Similarly, if I skip exon 3 and I will add, then I will get another different protein. So this kind of a splicing is what is called as the alternative splicing process. Clear, all of you? So here, this is what it is about the post-transcriptional modifications. Okay, so capping, tailing, and splicing. Also, you have alternate splicing, right? So we have just finished this transcription. So hope the class was useful, right? So we will meet with the next schedule. So thank you all. If you have any doubts, you can just post. So if you want the PDF, download the app in the description box with a link in the description box given below, and DM as PDF of this session. Okay, so you will get the PDF. Thank you all.